Hey everyone, welcome back to Window Seat Studies. So in last class, we were learning about different enzymes which are involved in histone methylation. So we first discussed about histone methyltransferases, the histone lysine methyltransferases to be specific. We are not discussing about arginine methylation yet. Then we discussed about the histone demethylases. Again, those are lysine specific. In that, we discussed about the LSD1 enzyme, which is lysine specific demethylase 1. And this enzyme, it works in context dependent manner. So depending on with which complex it is going to work, the substrate is decided. For example, if it works with androgen receptors, it demethylates the canine residue. And if it works with co-rest complex, it demethylates the K4 residue, H3K4 methylation mark is removed. Okay, so while discussing about histone demethylases, I forgot to mention another category of histone demethylases. Okay, and that other category is Jumonji domain containing demethylase. Okay. So in the lysine specific histone demethylases, those have two categories depending on which cofactor they require. The first one is FAD dependent. Okay, FAD dependent demethylases. So they require FAD as their cofactor, and the example we learned in yesterday's lecture was LSD1. The other category is the Jumonji domain containing demethylase, and instead of FAD, they need Fe2 as its cofactor, as well as they need 2-oxoglutarate. Okay, I'm not sure about the spelling. So they use 2-oxoglutarate as the other substrate. Okay, so for the functioning, this is also required. So Fe2 as well as 2-oxoglutarate is required for the Jumonji domain containing demethylase to work. So the first discovered Jumonji domain enzyme was JMJD2. Okay, so see LSD1, those have amino oxidase activity. Okay, so this LSD1 enzyme, it oxidizes the methyl group. Okay, methyl mark. Okay, but unlike this LSD1, those Jumonji domain, they fall under Fe2. Okay, Fe2 plus 2-oxoglutarate enzyme requiring category. So they hydroxylate the methyl mark. Okay, so the mechanism of functioning of those two enzymes is quite different. Okay, and that is why we are categorized them in two different groups. The first one has LSD1 as an example. This Jumonji domain containing demethylase has different enzymes such as JHDM1. Okay, Jumonji histone demethylase. Okay, JHDM, the JHDM2A, JHDM3A. So all of those three members of the JHDM demethylase, they act on the different substrates. So like JHDM1, it demethylates H3K36 monomethylation or dimethylation mark. Then JHDM2, it demethylates H3K9 monomethylation, dimethylation, or trimethylation mark, as well as the H3K. No, it doesn't work on H3K36. Okay, it only works on H3K9 methylation mark. So the other one, JHDM3A, it works on H3K36 trimethylation mark, or it can also work on. Oh, okay, it can also work on H3K9 trimethylation mark. Okay, so those are the three examples of Jumonji domain containing enzymes. So that's it for the demethylases. I forgot to discuss this one. That's why I added this enzyme in today's lecture. So with this, we are done with the lysine-specific histone methylation part. Let's so before moving on, let's do a quick recap. So here, this image is taken from a research article, and this research article actually discusses about the different different histone demethylases. So initially, it was it was thought that the just like the DNA methylation, histone methylation is also a permanent mark, or it is a stable mark which can't be reversed. But as soon as we discovered about the histone demethylases, we got to know that no, there this mark can be reversed. So this paper discusses about that, and it discusses about the Jumonji domain containing demethylases which come under the KDM family. So here you can see this is a nucleosome, okay, the octamer, and around which the DNA is wrapped. Okay, here you can see, and this is the H3 protein, the histone H3 protein, and this is the N-terminal tail of the H3 protein. So this is the histone tail of H3, and this is the histone tail of H4. Okay, so here you can see the different marks. There should this schematic showing the different mark. So here you can see this is the fourth residue, the H3K4. This is the ninth residue, H3K9. Again, this is the 27th residue, H3K27, and this is the 36th residue, H3K36. And this is the H4 protein. Here you can see this is the 20th residue. So this is H4K20. Yeah, there was one confusion. I need clarification about the H3K20 methylation mark. Okay, so certain research papers are showing that there's not enough information available about K20. Like there is ambiguity about whether it is. For me, I'm not yet sure about its function, like whether it's repressive mark or active mark. So I need a clarification on that topic. So, but there's a well-established fact that the H420 methylation, this is also done by SUV20 H1 or H2 enzymes that I discussed in the H3 K20 methylation mark. So like this is a well established fact. So I'll clarify about the H3 K20 methylation mark ASAP. So that's it. Yeah, we were discussing about the different methylation marks. So here you can see H3K4 methylation. This is done on either promoters of active genes or the enhancers. So, okay, so you can see that this is done on promoters or enhancers because the K4 methylation mark is an active mark. So we need it at the promoter or enhancer, the regulative elements of the active genes. Okay. Then again, the K9 methylation, H3K9 methylation, this is the hallmark of heterochromatin or repressed gene. And here you can see there's one green star, and it shows that it is also done in promoter or active genes. So this is worth noting that there are like certain exceptions to the rule, that like when we discussed about H3K9 methylation, we discussed that this is hallmark of heterochromatin. Like whenever there is a H3K9 trimethylation mark, you will automatically assume that it will be a repressive mark. Okay, but there is again an exception to this rule, that like tandem repeats or so in the tandem repeats or satellite DNA, there's some part which is active. Okay, and at that and that part is regulated by the H3K9 methylation mark. Okay, so sorry, yeah, H3K9 methylation mark. So this let me write this point. So H3K9 methylation mark. Okay, this is a repressive mark. Okay, generally it's repressive mark, but the exception is, okay, exception is that it also it also regulates some enhancer regions or the promoter regions. It regulates enhancer. Okay, wait. It also regulates enhancer regions which fall under tandem repeats or some satellite DNA. Okay. Okay, so this exception is worth noting. Okay, so like don't forget about it. So then the next mark is H3K27 mark. Here you can see the red stars. So it denotes So again, it denotes the denotes the heterochromatin or repressed genes. So we remember that it is associated with PRC2 complex silencing, the silencing which is mediated by polycomb repressive complex 2. And we will learn about this in detail when we will study the long non-coding RNAs as well as some other yeah, in the long non-coding RNAs, we'll learn about it. I'll explain the PRC2 complex, and again, we will learn about this in the polycomb while learning about polycomb group of proteins and the trithorax group of proteins. So the 27 is also related to the heterochromatin or repressed genes, but again, there's an exception to this rule that it can be present at the promoters or active genes. So this is an exception thing, okay? Then the K36 methylation, we learned that this is a very important and distinct because it is related to the transcription elongation. Then there is H4K20 methylation. It is also a repressive mark associated with heterochromatin, and there's exception also. So this slides, this schematic concludes all of the things that we have learned till now. So with this, we are done with histone methylation. Specifically the license specific histone methylation. So in license specific histone methylation, we learned about the certain specific marks, the certain methylation marks like those that we discussed now, the K9 methylation, K9 methylation, K4 methylation, K36, K27, and K20 methylation marks. Then we discussed about different enzymes like HKMTs, the set domain containing lysine methyltransferases or non-set domain containing ones. And then we discussed about the histone demethylases, the LSD one and as well as the Jumonji domain containing demethylases. So with this, we conclude the histone methylation series. So now let's learn about arginine specific methylation. Arginine specific histone methylation. Okay, so this arginine specific histone methylation, this is done by the enzymes known as PRMTs, protein arginine methyltransferases. So there are two types of PRMT, the type 1 PRMT and type 2 PRMTs. Okay, so why are those two? Why are they? What are the different characteristics of those two types? The type 1, this monomethylates the arginine, okay, as well as it does the asymmetric dimethylation. Okay, well, the type 2 PRMTs, it again monomethylates the arginine, so it monomethylates arginine, or it adds the symmetric dimethylation. Okay, so the asymmetric dimethylation, this is written as this. So RME dimethylation, but it is asymmetric. And if it is done in symmetric fashion, then it is written as this. Me dimethylation symmetric. So this is arginine asymmetric dimethylation and this is arginine symmetric dimethylation. So let's have a look at it. So this is the arginine residue, okay, and on which the both type 1 and type 2 residue no, type 2 PRMTs can work. If the type 1 PRMT catalyzes the reaction, then the after monomethylation the other methyl group is added in asymmetric fashion. You can see it is asymmetric. But if the type 2 PRMT works on it, then the methylation is done in the symmetric manner. Okay, the dimethylation is done in symmetric manner. So depending on whether this is it is okay, so the monomethylation mark is always an active mark. Okay, monomethylation mark is an active mark. But when it comes to the dimethylation part, depending on whether it's asymmetric or symmetric, it decides whether it will be the repressive mark or the active mark. So let's have a quick look at the mechanisms, okay, by which PRMTs, or say arginine methylation, regulates the transcription. Okay, so let's first learn about the H3R2 dimethylation. Okay, so when there is H3R2 dimethylation and it is symmetric, so this blocks the effector molecule of blocks the effector molecules of H3K4 methylation mark. Okay, and the H3R2 dimethylation, if it is asymmetric, it does not let the enzymes add the H3K4 methylation mark. Okay, so like both of those, so consider both of those, the asymmetric or symmetric mark. Both of those, they are either directly inhibiting the addition of the H3K4 methylation, or they are they are inhibiting H3K4 methylation mark to work. Okay, so like blocking the effector molecules would like there will be the methylation of the K4 lysine residue, fourth lysine residue, but now the effector molecules, like whatever it is going to recruit at that site, all of those will not be they will not settle down, they will not form the scaffold over there, and now it is inhibiting the H3K4 activity. So both of those marks, either symmetric mark or asymmetric mark, they are resulting in the inhibition of active marks, and that is why those are repressive mark. Okay, so the dimethylation of the second arginine residue on H3 protein, whether it is symmetric or asymmetric, it is an repressive mark. Okay, so this was the first example. The second example is of H4R3 dimethylation. Okay, so if this H4R3 dimethylation is asymmetric, okay, no, if it is symmetric, then this mark, this is a hallmark. This mark provides a docking site for DNMT3A. Okay, for DNMT 3A. So I hope you remember about DNMT3A. This is de novo methyl transferase. So this is associated. So this is the symmetric one. So this is associated with PRMT5. Okay. Oh yeah, I forgot to mention the examples of those PRMTs. The type 1 PRMT, which does the monomethylation or asymmetric dimethylation, has the example like PRMT1 or CARM1, while the type 2 has the example PRMT5. Okay, and it adds the symmetric dimethylation. Okay. Hmm. So the type 2, it does the symmetric dimethylation. Okay, so this is the symmetric dimethylation of third like arginine residue, and that is why it is done by PRMT5. So the activity of PRMT5 is related with the DNA methylation. Okay, why? Because this mark provides a docking site for the de novo methyl transferase DNMT3A, and so now due to the DNMT3A binding, the DNA will get methylated, and that is why we can say that this H4R3 is the repressive mark. Okay. Okay, so this is how the methylation, those methylation marks could be either repressive mark or active mark. There's other example of the arginine methylation, which can be active mark, but it all depends on whether it is the symmetric methylation or asymmetric methylation. Okay, in this example, both of them are repressive mark. They are not letting this H3K4 methylation mark, which is an active mark, to work, but the mechanism is different. Like this symmetric one is blocking the effector molecule, while the asymmetric one is not letting the K4 residue to get methylated. Okay, so this is how the arginine methylation is also important. So with this, we are done with the histone methylation part. So should we do a quick recap? Okay, let's do it. So in the histone methylation part, we learned that the histone methylation. So I'm summarizing all the 5 to 6 lectures that we were that we in which we were discussing about histone methylation. So histone methylation means addition of CH3 group on either lysine residue or arginine residue. On lysine residue, it is done by HKMTs. Okay, on arginine residue, it is done by PRMTs. The demethylases, the HDMs for lysine residues are different from the HDMs of the arginine residues. Okay, so so in the lysine residue, there the histone demethylases are either FAD active. Okay, the example is LSD1, or they require Fe2 as well as 2-oxoglutarate to work, and the example is JMJD2, Jumonji domain containing demethylases. The HKMTs are again of two types, the set containing HKMTs and the HKMTs which do not have set domain. Okay, the set containing HKMTs are of different type, the set 1, set 2, set 7, set 9, then MLL complex, SUV39H1, SUV20H1 and H2, again EZH complex, enhancer of zest. So those are all examples of set domain containing histone methyl transferases. But there is one example which does not have set domain, and that is DOT1L. And this enzyme methylates the 79th lysine residue. Okay, H3. So it adds the H3K79 methylation mark. Okay, this is very important because it resides in the core histone. In the arginine, there are two PRMTs, the type 1 and type 2. Type 1, both of those monomethylates arginine, okay. The type 1 does the asymmetric dimethylation, while type 2 does the symmetric dimethylation. Okay, the HTMs. Okay, in the arginine, there are no histone demethylase as such. Like there is in those we saw that they either oxidizes the methyl mark, or they can hydroxylate the methyl mark. But in the arginine methylation, there are no such histone demethylase. Rather, there was seen that there is an enzyme known as PAD4. This is peptidyl amino uh, amino deaminase. Okay, 4. So this PAD4 enzyme, this is actually a deaminase. This does not demethylate the arginine, but it can replace this methylated arginine with the citrulline. Okay, so if you do not know, citrulline is the this is the non-standard amino acid. Okay, so like we can actually call this as the demethylation if this RME is replaced by the arginine, right? But this RME is actually replaced by citrulline, and we do not like as of now we do not know any any mechanism by which this citrulline is removed and it is replaced by arginine. So we do not actually consider this PAD4 as the arginine demethylase as of now, because we do not have sufficient information on this. But yeah, there was a paper which I guess discussed about JMJD6. Yeah, JMJD6. I'm not sure if it is something else. I'll post it in the comment section. So it discussed that JMJD6 demethylates the arginine, like it replaces arginine methylated arginine with arginine somehow, but this is not yet confirmed. Okay, so a lot of investigation has to be done on this part yet. So for now, let's just assume that there are no histone demethylases, but there is an enzyme PAD4 which can replace methylated arginine with citrulline. Okay, so this concludes the histone methylation part. Okay, so this was about the enzymes, enzymes which are involved in histone methylation. Then again, we learned about the methylation marks. First, let's learn about the lysine specific methylation mark, the H3K9 methylation, H3K27 methylation mark. Then we learned about H3K4 methylation mark, H3K36 methylation mark, as well as the H4K 20 methylation mark. Okay, so all of those different methylation mark we have learned about it. The H3K9 methylation mark is hallmark of heterochromatin. H3K27 is also a repressive mark. It is related to PRC2 complex silencing. H3K4 methylation mark is an active mark. H3K36 methylation mark is related with the transcription elongation. So H3K4 methylation mark is seen at the transcription start site, while this is seen at the 3' end of the transcribing genes because it is related to the transcription elongation. H3K420 mark is again active mark, but it can be repressive mark. No, it is actually repressive mark, but it is active mark if it is mono or dimethylation. Like there are always exceptions to those rules. Okay, so those were the license specific methylation mark, and then in the arginine we learned about H3R2 dimethylation mark, symmetric or asymmetric. Then H4R3 methylation mark, symmetric or asymmetric. Okay, so that's it. With this, we are done with histone methylation, and in next lecture we will learn about the other histone modification, that will be histone phosphorylation, and after that we will also learn about histone ubiquitination. And with this we will be done with histone modification part, and once we are done with histone modification, we'll start a new topic, and that is a very interesting one. Personally, it's favorite. So I like that topic, and that would be long non-coding RNAs. So that's it for today. Till then, see you. Bye bye.